today we are going to study about one of the most important bacteria in the microbiology, which is called Mycobacterium tuberculosis. So let's start. So talking about the important points regarding Mycobacterium tuberculosis, the first point is that it is acid-fast bacilli, means that it can't be stained by ordinary gram staining, and we have to use another staining called G-Nielsen staining. G-Nielsen staining. It can't be stained by ordinary gram stain due to the presence of mycolic acid in its cell wall. And this mycolic acid retain the carbol-fission stain, although we have treated it with the ethanol hydrochloric acid, but after its treatment with ethanol hydrochloric, it still retains the carbol fusion stain, and thus it is called acid-fast bacilli because it is fast to the acid. Fast means it can resist the acid like hydrochloric acid. The second point, that it is an obligate parasite. The third point, that it is very slowly growing, having a doubling time of about 18 hours. It is non-encapsulated, non-motile, non-spore forming. So these are some of the important points regarding the Mycobacterium tuberculosis. Now we shall study about the difference between the virulent strains and the non-virulent strains. So in order to distinguish between the virulent strains and non-virulent strains, we must keep in mind the two important points. First, that the virulent strains consist of cod vector, which is chemically trehalose dimycolate, and is required for the survival of Mycobacterium tuberculosis. The other point is that on microscopy it appears like a serpentine cord-like pattern. It gives serpentine cord-like pattern, as you can see in this slide, in this picture. It's about serpentine cord-like patterns of Mycobacterium tuberculosis. Talking about its hardness, so they are one of the most hard bacterium in the microbiology. They are resistant to acids in the stomach. They can't be killed by the acids in the stomach. They are resistant to acid, and they are also resistant to dehydration. Now talking about its transmission epidemiology, so humans are the natural reservoir. Although cattle can be infected, but they are not the reservoir for humans, i.e. they cannot transmit this bacterium to the humans. This bacteria can only be transmitted from person to person by the respiratory droplets produced during coughing, sneezing, etc., by the dust-containing organism, by fomites, and by the food which are handled by the infected person having Mycobacterium tuberculosis. So talking about these virulent specters and exotoxin, endotoxin, also they produce no exotoxin, endotoxin. They produce some of the virulent specter like tuberculosis necrotizing toxin, early secreted antigen 6, and exported replicated protein. Talking about one of the tuberculosis necrotizing toxin, so it degrades the nicotinamide dinucleotide, which is one of the important cofactor for the survival of the macrophages. So degrading this will cause death of the macrophages. The second important virulent specter is the early secreted antigen 6, which reduces the innate immunity against the Mycobacterium tuberculosis and other organism by reducing the secretion of gamma interferon. The third one is the exported replicated protein, which prevent the fusion of lysosome with the phagosome phagosome containing Mycobacterium tuberculosis and the lysosome containing uh, different type of enzymes which cause death of the Mycobacterium tuberculosis. So preventing their fusion increase the survival rate of Mycobacterium tuberculosis and cause disease. Now coming towards pathogenesis, how they cause disease. So once the bacteria enter the lungs, they are uh, engulfed by the bacteria present in the alveoli called alveolar macrophages and start inflammatory reaction against this bacteria to cause one of the important disease called primary tuberculosis. Primary tuberculosis. And this primary tuberculosis consists of two phases: in initial phase and the tubercular or late phase. Talking about the initial phase, it's characterized by the parenchymal exudative lesion, which is called this exudative lesion. Parenchymal exudative lesion is called GHON FOCUS, and it is mainly caused by the acute inflammatory response against the Mycobacterium tuberculosis. This exudate consists of, for example, proteins, different types of cells like neutrophils, etc. In contrast to the transudative, we do not contain proteins and neutrophil cellular substances. This GHON FOCUS along with the draining lymph nodes, this is called GHON complex. For example, this is the GHON FOCUS and the draining lymph node which drain this GHON FOCUS. These both things are called GHON complex. Now, talking about the late phase, also called tubercular phase. So this is characterized by the productive or granulation lesion, which consists of the Mycobacterium tuberculosis in the giant cells. This giant cells is also called Langerhans giant cells, if you will see in the picture. And this Langerhans giant cells are surrounded by the epithelioid cells. What are epithelioid cells? They are the activated macrophages. They are the activated macrophages having a shape of about like epithelial cells. And this Langerhans giant cells surrounded by the epithelioid cells leads to the formation of tubercle. And what is tubercle? It is a granuloma surrounded by fibrous tissues having a central caseation necrosis. Caseation necrosis, as you can see in this picture. This is a lung. This is a GHON complex. Infection with the Mycobacterium tuberculosis enter the lungs to the trachea and cause formation of the tubercle consisting of lymphocytes, as you can see, epithelioid macrophages, as you can see, consisting of Mycobacterium tuberculosis, multinucleated giant cells consisting of Mycobacterium tuberculosis, and the central caseation necrosis. Caseation means that it look like cheese when seen on naked eye. Now, if it causes primary tuberculosis, as we have already discussed, and leads to the formation of tubercle, then there are two chances of the tubercle. It can be arrested, i.e., no more growth in the tubercle, which then heals by the fibrosis and calcification. And this tubercle, i.e., GHON complex, when heals by calcification, then it is called Ranke's complex if it heals by calcification. The other chance is that this tubercle breaks down, and this can lead to one of the four important phenomena. The first one, which occurs in about 10% of the infected people, that can lead to active tuberculosis, which can lead to death, or if treated in a good manner, it can lead to the healthier person. The other chance that it can spread to all the lung, causing progressive lung diseases, and this progressive lung disease can in turn leads to death. The third chance is that it can enter the blood to cause severe bacteremia. From the alveoli, it can enter the capillaries, and from the capillaries, it can go to the systemic circulation. It can lead to severe bacteremia, which is called miliary tuberculosis or disseminated tuberculosis. Disseminated tuberculosis. The fourth chance is that it can, in 90% of the infected person, it can spread to lymph nodes and bloods to and seed the other organs like liver, lung, bones, meninges, and become latent or dormant in these organs. Now, in the next slide, we will study about the dormant stage. So, in the dormant or latent stage, it can be reactivated. This latent or dormant tuberculosis can be reactivated in the adults and elders, mostly in the alcoholics, in the smokers, in the immunocompromised, having AIDS like diseases. This can be reactivated and cause secondary tuberculosis, which is of two types. The first one is the pulmonary tuberculosis, which causes disease in the lungs, and the third one is the extrapulmonary tuberculosis, which is of different types like intestinal, renal, etc. tuberculosis. And we will discuss tuberculosis. So we will discuss about the clinical finding caused by the Mycobacterium tuberculosis in the next slide, in the next lecture. So stay tuned and thank you. Jazakallah.